Hello everyone, my name is Priyanka. My qualification is MSc B.Ed and CSIR. Today's topic is meiosis and its significance. You know, you all of you must know about cell division, that is mitosis and meiosis. Mitosis happens in vegetative cells for the increasing the number of cells, but meiosis happens in the reproductory cells when reproduction is going to happen. Why? Because after meiosis, one cell is going to form four cells. So, and the chromosome number is going to reduce to half in the daughter cells. So, if parents will have 46 chromosomes, then after meiosis, cell will form gametes, and one gamete is going to have 23 chromosomes, exactly half the number of chromosomes of the parent cell. So, this is the importance of meiosis. And one more important thing is when sexual reproduction happen, fusion of will happen. So the chromosome number should be half in those gametes. Then when they will fuse and form one zygote, the chromosome number will be same as of their parents. So this is the importance of meiosis. It will it will continue the number of chromosomes from one generation to other generation. The chromosome number is not going to increase, it will remain same. The second thing is variation means difference among the species of among the individuals of same species. You know that if one couple has two or three children, all of them will have many things similar to each other, but many things different from each other. So the siblings of same species which have difference in among them, that is called as variations. So by this process of meiosis, these variations comes in the individuals, and these variations come due to phenomenon of crossing over which happens in the meiosis and the genetic recombination. So, and both variation, crossing over and genetic recombination is very much important for evolution of organism. What are stages of meiosis? You know that whenever any cell divides, it is going to prepare itself for division, that is called interphase. And the other phase where actually cell divide, that is divided into four phase: prophase, metaphase, anaphase and telophase. But in meiosis, this division happens in two stages because there is going to formation of four cells. So in one division, one cell will form two cell, and in other division, two cell will form four cell. So cell division is here some long from the mitosis. But before cell division, that is called interphase or preparatory phase, it is also divided into three phases: G1 phase, S phase and G2 phase. I have discussed this topic in very detail in my slide mitosis and interphase. You can watch that top slide for the actual understanding of interphase. I am just explaining it here for the understanding. G1 phase is preparatory phase, and here cell is going to physically will increase itself and form protein and molecules needed for cell division. In S phase, inside one chromosome, from one strand of DNA, there is going to formation of another strand of DNA. It means that if in G1 phase, cell will have one chromosome, and inside one chromosome one strand of DNA. So this is G1 phase. What will happen in G2 phase? Cell will have one chromosome like before, but number of DNA is going to will be doubled. You can see the number of chromosome is same in G1 phase and S phase, but the strand of DNA is doubled here. So amount of DNA is exactly doubled in S phase. If here it is 2C amount of DNA 2C, then here it is 4C, exactly double, because here one strand is present, here two strand is present inside one single chromosome. That is difference between G1 phase and S phase. And G2 phase is another gap phase where cell form more protein needed for cell division, and from G2 phase cell enters in mitosis or meiosis. In meiosis, G2 phase is very short. It is either absent or very short. It is not very much required. So, in meiotic division, I have told you that meiotic division is a long division. First, one cell will divide in two cell, and two cell will divide in four cell. So, in first meiotic division, there is four the stages of cell division is formed, that is prophase, metaphase, anaphase and telophase. And again in second meiotic division, all these four stages will again going to be present. So prophase, metaphase, anaphase, telophase for first meiotic division, and prophase, metaphase, anaphase, telophase for second meiotic division. But in second meiotic division, prophase is actually either absent or very short. Here prophase also is divided into 5 more phases, because here prophase is very important, because all the interesting characters of meiosis is going to happen in prophase, that is crossing over, genetic recombination. So for the convenience of understanding, prophase is divided into 5 phases, that is leptotin, zygotin, diplotin, dikinesis. Leptotin, zygotin, pachitin, diplotin and dikinesis, 5 phases, and each phase is very important. What happens in leptotin? In leptotin, chromosomes look like thread-like structure or beads on string structure, and nuclear membrane is intact, and it will look like the noodles in a soup bowl. If you consider nucleus as a soup bowl, then the chromosome will look like the noodles inside a soup bowl. See, you can see the picture of leptotin. Here this is the one chromosome, and this is the other homologous chromosome. You all must know what is homologous chromosome. Homologous chromosome are chromosomes which have similar type of gene in same sequence. It means that if one gene have two allele, like for example, if the height or gene of height, that is have dominant allele for T, it will have recessive for T here at the same locus. That's why they are homologous chromosomes, and one will come from male and one will come from female. You should be clear about what are homologous chromosomes, then you can understand the other part of the prophase. Homologous chromosomes have two alleles of similar gene and in a similar sequence. One homologous will come from mother and one homologous will come from father. The concept of homologous chromosome only occurs in meiosis. It does not describe in mitosis, because there is no need of homologous chromosome in mitosis. Homologous chromosomes are present in mitosis. It is not that they are disappeared, but they divide randomly. They don't come each other, or they don't have to cross over with each other. Here it is important to tell you that what are homologous chromosomes and how they come to close to each other, and what they do with each other, and how they separate from each other. That's why I am telling you what is homologous chromosome. In leptotin, they are just lying anywhere inside the nucleus, homologous chromosome. But in zygotin, they start coming close to each other and start pairing, and that pairing is called synapsis. Synapsis is a pairing like zipper like fashion. For one part they will be close and other part they will be separate, like a zipper. When we close the zipper, what we'll see, one part is going to be closed. See, synapsis is very much important in zygote. That's why it is called zygote, because two homologous chromosomes are coming near each other. And pairing is allowed only at one reason. It doesn't mean that whole chromosome will pair side by side. No, the pairing will occur at one point of two chromosomes. You can see it here, you can understand it. Here 4 strands of DNA are shown. 4 strands of DNA have been shown in the picture like this. If one chromosome is this, and here 2 strands of DNA is here in one, and this is other homologous chromosome, and it will also have 2 strands of DNA. Then here only strands of DNA is shown. It means that this is one chromosome having 2 strands, and this is other homologous chromosome having 2 strands. You should not be confused what is showing here. You should know, clearly understand, these are 2 strands of DNA, 2 from one chromosome and 2 from other chromosome. This is very much important, otherwise you will be confused at later stages. So you can see 1 is 2 pairing here at this point, 2 and 3 are pairing at this point, and 3 or 4 are pairing with each other at this point. So they pair at only one point along their length. And then comes patching, third stage of prophase 1. But it's a very important stage, because in this stage you can see both the strands of chromatids will be visible. It means that you can here see only one chromosome, only one homologous chromosome. This is other homologous chromosome. This is 1 and this is 2. You cannot see the strands of DNA inside it clearly. But in patching, you can see one chromosome is having 2 strands very clearly. Both the strands are visible only in patching strand. These are 2 strands of DNA, and they are visible in patching strand, and that's why they are called bivalents, because one strand will form one chromatid. One strand of DNA will look like one chromatid, and the other will look like other chromatid. These are called bivalent. It is not visible in zygote and leptotin. The bivalent is visible only in patching strand, because here one strand will form one strand of DNA is looking as one chromatid, and other of strand of DNA is looking as other chromatid. So 4 strands are clearly visible here. That's why they will call as tetrad. Each chromosome in a bivalent at this stage has 2 chromatids, as a result of which a bivalent really consists of 4 chromatids and is called as tetrad. This is called bivalent. Both the homologous chromosome form bivalent, and they will have 4 chromatids, 1, 2, 3 and 4. So bivalent is made up of 4 chromatids. This is the importance of patching. And the other important thing of patching is crossing over. The process of crossing over happens. The crossing over What is crossing over? Crossing over means that one part of this chromosome will exchange the genetic material with this part of the other homologous chromosome. It means that one chromatid will exchange some amount of DNA with the other chromatids. This is called crossing over. It is very important because during the process of crossing over, one allele can be transferred from this chromatid to this chromatid. It means that here, if the allele A is present and here the dominant form of this allele A is present, so during crossing over, it may happen that this small a can come here on this chromosome and this capital A can be transferred to this other. So by the process of crossing over, one allele can be transferred to the respective homologous chromosome and the other allele of that chromosome will come to this chromosome. It means that it is happen like this. A will go to the here and from here A will go to the other chromatid. This is called crossing over and this is very much important. And this crossing over happens by the crossing over genetic recombination. Recombination means formation of new combination will happen. So if on this chromosome, if the here the gene number 1 is present, A is present, gene B is present, gene C is present, so after and here A is present, B is present, C is present. So after crossing over, it may happen that the A will come here, A will come here and the allele A will come here, capital A will come here. And so what happens after crossing over, the place of allele will exchange with each other and then new combination is appearing. Now the chromosome will look like this chromatid will have all the dominant and this chromatid will have all the recessive allele, and previously the one recessive will A was here and the capital was here. So this is the importance of crossing over. New combination will appear. What happens in diplotin? The point where two chromosomes will cross with each other or exchange their genetic material, that is called chaismata. They look like the point of contact between two chromosomes, chaismata. So what happens, these chaismata or points of contact will go towards the periphery or ends of the chromosome. They will start disappearing. And the final stage of prophase 1 is dikinesis. It means that chaismata are mainly terminal. It means they are only found in terminal positions of chromosome, and now the nuclear membrane will start disappearing. Why? You have the inside a cell the nuclear membrane is found, and inside the nuclear membrane the chromosome is found. But at the stage of dikinesis, this nuclear membrane will start disappearing. Why? Because after dikinesis, when nuclear membrane will disappear, the spindle apparatus, it means that one kind of protein is going to form here and one kind of, and it will form spindle apparatus. This spindle apparatus is a protein and it will attach itself with the centromere of the chromosome, and they will separate the chromosomes from each other, so can they go to the opposite pole and they can divide the chromosome. So for that reason nuclear membrane is going to disappear. see, you can see here, prophase first is prophase first is shown here. Two chromosome, the blue and pink are homologous chromosome. Now in metaphase 1, after prophase, first metaphase will come. And what is importance of first metaphase? Importance is that the chromosome will arrange themselves, two homologous chromosomes arrange themselves side by side, one blue and one pink, that are homologous, and other pink and other blues, that are also homologous. So it means that, and spindle apparatus, which is a proteinaceous structure, and it will form spindle. Spindle means string or rays. They will attach themselves with the one centromere of one homologous chromosome, and the other spindle apparatus will attach with the centromere of other homologous chromosomes. It means that if these are homologous chromosome with two chromatids, and you know that chromatids are attached at the point of centromere, where the two chromatids of one chromosomes are attached, that is called centromere. This is centromere. So spindle apparatus will attach with the centromere of one homologous chromosome, and the other apparatus will attach the centromere of other homologous chromosome side by side. The homologous chromosome will arrange themselves side by side, and only one will attach with the one side of spindle apparatus, and other one will attach themselves with the other side of spindle apparatus. So after metaphase, when anaphase will come, the one pair of one homologous will go from one pair, one homologous chromosome will go to one pole, and the from other homologous chromosome will go to other pole. This separation is non-disjunctional. Non-disjunctional means only, you can see here in metaphase, only blue is going to one pole and only pink is going to the other pole. Here from second pair, only blue is going to one pole and only pink is going to one pole. This is not equal division. This is non-disjunctional division. I hope you have understand. It's very much important. There is a very profound difference between the metaphase of mitosis and metaphase of meiosis. That is, in metaphase of mitosis, the chromosome arrange themselves like this. The blue one will arrange like this, and the pink one will arrange like this. They arrange themselves in one by one, not side by side like in meiosis, metaphase 1 of meiosis. They will arrange in one by one. So what happen will, one spindle apparatus will join with both the sides of one homologous chromosome. This one will join both the side of spindle apparatus, and this pair will also join from both the sides of spindle apparatus. So in mitosis, this is for mitosis, every pair of homologous chromosome will attach themselves with each side of spindle apparatus, and when they will divide, the centromere splits from here. What will happen that when centromere will split from here, one will go to one cell and one chromatid will go to other cell. In the pink one also, one will go to one cell and the other will go to other cell. This happens in mitosis. This is very much important point. You should be clear about this, like a crystal clear water. See, from in mitosis every homologous chromosome is going to one cell. It will divide in mitosis every homologous chromosome is going to divide equally. That's why mitosis is called as equational division. But it does not happen in meiosis. In meiosis the homologous chromosome arrange themselves as metaphasic plate side by side, and only one centromere will attach themselves with the one side of spindle apparatus. So when the nucleus will divide at telophase, only one type of chromosome is present inside the one nucleus or inside one cell. You can see here one blue from this pair and one pink of this pair will come in one cell, and one pink of this and one blue of this will come inside this cell. They are not same, they are different. Both the cell will have different type of chromosome. The genetic material will be little bit different in both the cells after meiosis, but in mitosis the genetic material is same. It is equal division, and it's very much important character to understand. It happens in anaphase. Here I have written that the meiosis is called a reductional division. Why? Because the number of chromosome is going to reduce to half. And mitosis is called a equational. I have written the difference because it's very important and it's asked in many type of exams, that what is difference between reductional or equational division, who is the reductional division, mitosis or meiosis, and what is the equational division, mitosis or meiosis. You should be clear about this point. The second division of after anaphase, telophase happens, and after telophase, second meiotic division will start. And second meiotic division is similar to mitosis. It means it is simply division of chromosomes, simply division of cells into two cells, like mitosis. It's very much similar to mitosis. You can see here in telophase 1, when the cell is divided into two cells, then what will happen is second phase of meiosis. In second phase of meiosis, one half of the chromosome will go in one cell and half will go in other cell, and this will happen with this also. Same thing will happen with this nucleus. So four cells will going to form after meiosis like this. This one will go in one cell and this half will go in other cell. So thanks for watching. This was my today's topic. If you like my content, then subscribe and do comment in my comment box.